students, let's start with chapter 7th of Harper's Illustrative Biochemistry, which is Enzymes Mechanism of Action. First, let us see the objective of the chapter. After studying this chapter, you should be able to describe the structural relationship between specific B vitamins and certain coenzymes, outline the four principal mechanistic strategies employed by enzymes to catalyze chemical reactions, explain the concepts of the lock-in key and induced fit models for enzyme-substrate interaction and how the latter accounts for the dynamic nature of enzyme catalysis. Outline the underlying principles of enzyme-linked immunoassays. Describe how detecting and measuring the activity of many enzymes can be facilitated by coupling with appropriate dehydrogenase. Identify proteins whose plasma levels are used as biomarkers for diagnosis and prognosis. Describe the application of restriction endonuclease and of restriction fragment length polymorphisms in the detection of genetic diseases. Illustrate the utility of site-directed mutagenesis for the identification of amino acyl residues that are involved in recognition of substrates or allosteric effectors or in the mechanism of catalysis. Describe how affinity tags can facilitate purification of a protein expressed from its clone gene. Discuss the events that led to the discovery of the RNAs can act as enzymes and briefly describe the evolutionary concept of an RNA vault. So first starting with biomedical importance. Nobel laureate 1946 James Sumner defined an enzyme as a catalyst of high molecular weight and biological origin. Enzymes catalyze the chemical reactions that make life on Earth possible, such as the breakdown of nutrients to supply energy and biomolecular building blocks. The assembly of those building blocks into proteins, DNA, membranes, cells, and tissues, and the harnessing of the energy to power cell motility, neural function, and muscle contraction. Almost all enzymes are proteins. Notable exceptions include ribosomal RNAs and a handful of RNA molecules imbued with endonuclease and or nucleotide ligase activity known collectively as ribozymes. Many pathologic conditions are the direct consequence of changes in the quantity or in the catalytic activity of key enzymes that result from genetic defects, nutritional deficits, tissue damage, toxins, or infection by viral or bacterial pathogens, example Vibrio cholerae. Thus, the ability to detect and to quantify the activity of specific enzymes in blood, other tissues, fluids, or cell extract provides information that enhances the physician's ability to diagnose many diseases. In addition to serving as the catalyst for all metabolic processes, the impressive catalytic activity and substrate specificity of enzymes enables them to fulfill unique roles in human health and well-being. The protease renin, for example, is utilized in the production of cheeses, while lactase is employed to remove lactose from milk to benefit lactose intolerant individuals. Proteases and amylases augment the capacity of detergents to remove dirt and stains, while other enzymes can precipitate in the stereospecific synthesis of complex drugs or antibiotics. So in the figure, we can see the planar representation of 3-point attachment of a substrate to the active site of an enzyme. Although atoms 1 and 4 are identical, once atom 2 and 3 are bound on the complementary sites on the enzyme, only one atom can bind. Once bound to an enzyme, apparently identical form atoms thus may be distinguishable, permitting stereospecific chemical change. Enzymes are efficient and highly specific catalysts. The enzyme that catalyzes the conversion of one or more compounds substrate into one or more different compounds products generally enhance the rates of corresponding non-catalyzed reaction by factors of 10 to the power 6 to 10 to the power 9 or even more. Although some enzymes become modified during catalysis, these transient alterations are resolved in the course of the reaction being catalyzed. In addition to being highly efficient, enzymes are also extremely selective. Typically, the catalysts used in synthetic chemistry are specific for the type of reaction catalyzed and will act on any compound containing the relevant functional group. Enzymes, however, are generally specific for a single substrate or even a single stereoisomer thereof. For example, D but not L sugar, L but not D amino acids, or a small set of closely related structures. Since they bind substrate through at least 3 points of attachment as we have seen in the figure, enzymes also can produce chiral products for non-chiral substrates. For example, reduction of non-chiral substrate pyruvate by lactate dehydrogenase produces exclusive the L-lactate, not the racemic mixture of LD and L-lactate. The exquisite specificity of enzyme catalyst imbues living cells with the ability to simultaneously conduct and independently control a broad spectrum of biochemical processes. Enzymes are classified by reaction type. Some of the names of the enzymes first described in the earliest days of biochemistry persist in use to this day. Examples include pepsin, trypsin, and amylase. Earlier biochemists generally named the newly discovered enzymes adding suffix "ase" to the descriptor of the type of reaction catalyzed. For example, enzymes that remove elements of hydrogen H2 or H minus plus H plus generally are referred to as dehydrogenases. Enzymes that catalyze hydrolyzed protein are as proteases, and enzymes that catalyze rearrangement in configuration as isomerases. In many cases, these general descriptors were supplemented with terms indicating the particular substrate on which the enzymes act. Xanthine oxidase, its source, pancreatic ribonuclease, its mode of regulation, hormone sensitive lipase, or a characteristic feature of its mechanism of action, cysteine protease. When needed, alphanumeric designators can be added to identify multiple forms or isoenzymes of an enzyme. Example, RNA polymerase 3, protein kinase C beta. As more enzymes were involved, these early naming conventions increasingly resulted in inadvertent designation of some enzymes by multiple names or the assignment of duplicate names to enzymes exhibiting similar catalytic capabilities. To address these problems, the International Union of Biochemistry (IUB) developed a system of enzyme nomenclature in which each enzyme has a unique name and code number that identify the type of reaction catalyzed and substrates involved. These enzymes are grouped into the following six classes: oxidoreductases, enzymes that catalyze oxidations and reductions; transferases, enzymes that catalyze transfer of moieties such as glycosyl, methyl, or phosphoryl groups; hydrolases, enzymes that catalyze hydrolytic cleavage of CC, CO, CN, and other covalent bonds; lyses, enzymes that catalyze cleavage of CC, CO, CN, and other covalent bonds by atom elimination, generating double bonds; isomerases, enzymes that catalyze geometric or structural changes within a molecule; ligases, enzymes that catalyze the joining together ligation of two molecules in reactions coupled to the hydrolysis of ATP. The IUP name of hexokinase is ATP dehexose 6 phosphotransferase EC2.7.1.1. This name class identifies hexokinase as the member of class 2 transferases, subclass 7 transfer of a phosphoryl group, subclass 1 alcohol is the phosphoryl receptor, and hexose 6 indicates that the alcohol phosphorylated is on carbon 6 of a hexose. While EC numbers have proven particularly useful to differentiate enzymes with similar functions or similar catalytic activities, IUP names tend to be lengthy and cumbersome. Consequently, hexokinase and many other enzymes commonly are referred to using their traditional, albeit sometimes ambiguous names. Prosthetic groups, growth factors, and coenzymes play important role in catalysis. Many enzymes contain small molecular or metal ions that participate directly in substrate binding or in catalysis, termed prosthetic groups, cofactor, and coenzymes. They extend the repertoire of mechanistic capabilities beyond those afforded by functional groups present on the amino acid side chains of peptides. Prosthetic groups. Prosthetic groups are tightly and stably incorporated into a protein structure by covalent bonds or non-covalent forces. Examples include pyridoxal phosphate, flavin mononucleotide FMN, flavin adenine dinucleotide FAD, thiamine pyrophosphate, lipoic acid, biotin, and transition metals such as Fe, CO, Cu, Mg, Mn, and Zn. Metal ions that participate in redox reactions generally are bound as organometallic complexes such as the prosthetic groups heme or iron sulfur clusters. Metals may facilitate the binding and orientation of substrates to the formation of covalent bonds within the reaction intermediates CO2 plus by coenzyme B12, or by acting as Lewis acids or bases to render substrates more electrophilic, electron poor, or nucleophilic, electron rich, and hence more reactive. Cofactors associate reversibly with enzymes or substrates. Cofactors serve functions similar to those of prosthetic groups and overlap with them. The major difference between the two is operational, not chemical. Cofactors bind weakly and transiently to their cognate enzymes or substrates, forming dissociable complexes. Therefore, unlike associated prosthetic groups, cofactors must be present in the surrounding environment to promote complex formation in order for catalysis to occur. Metal ions form the most numerous class of cofactors. Enzymes that require metal ion cofactor are termed as metal activated enzymes to distinguish them from metalloenzymes, for which bound metal ions serve as prosthetic groups. It is estimated that one third of all enzymes fall into one of these two groups. Many coenzymes, cofactors, and prosthetic groups are derivatives of B vitamins. The water soluble B vitamins supply important components of numerous coenzymes. Nicotinamide is a component of the redox coenzymes NAD and NADP. Riboflavin is a component of redox coenzymes FMN and FAD. Pantothenic acid is a component of acyl group carrier coenzyme A. And its pyrophosphate thiamine participates in the decarboxylation of alpha keto acids, while folic acid and cobamide coenzymes function in one carbon metabolism. In addition, several coenzymes contain the adenine ribose and phosphoryl moieties of AMP or ADP. See figure 7.2. In this figure, we can see the structure of NAD plus or an NDP plus. So, this is the figure. Now coming back to coenzymes serve as substrate shuttles. Coenzymes serve as recyclable shuttles that transport many substrates from one point within the cell to another. The functions of these shuttles is twofold. First, they stabilize species such as hydrogen atoms FeTH2 hydride ions NADH that are too reactive to persist for any significant time in the presence of water and oxygen or the organic molecules that permeate cells. Second, they increase the number of points of contact between the substrate and enzyme, which increases the affinity and specificity with which small chemical groups such as acetate coenzyme A, glucose UDP, or hydride NAD plus are bound by their target enzymes. Other chemical moieties transported by coenzymes include methyl groups folates and oligosaccharides dolicol. Catalysis occurs at the active site. And importantly, early 20th century insight into the enzymatic catalysis sprang from the observation that the presence of substrate renders enzyme more resistant to the denaturing effects of elevated temperatures. This observation led Emil Fischer to propose that enzymes E and a substrate S form an enzyme substrate ES complex whose thermal stability is greater than the enzyme itself. This insight profoundly shaped the understanding of both the chemical nature and kinetic behavior of enzyme catalysis. Fischer reasoned that the exquisitely high specificity with which enzymes discriminate their substrates when forming an ES complex was analogous to the manner in which a mechanical lock distinguishes the proper key. The analogy to enzyme is that the lock is formed by a surface accessible cleft or pocket in the enzyme called active site. As implied by the adjective active, the active site is much more simply a recognition site for binding substrates. It provides the environment wherein chemical transformation takes place. Within the active site, substrates are brought into close proximity with one another in optimal alignment with the cofactors, prosthetic groups, and amino acyl side chains that participate in catalyzing the transformation of substrates into products. As we can see in this figure 7.3, two-dimensional representation of dipeptide substrate glycylation bound with the active site of carboxypeptidase A is shown. Catalysis is further enhanced by the capacity of the active site to shield substrate from water and generate an environment whose polarity, hydrophobicity, acidity, or alkalinity can differ markedly from that of the surrounding cytoplasm. Enzymes employ multiple mechanistic strategies to facilitate catalysis. Enzymes use combination of four general mechanistic strategies to achieve dramatic enhancement in the rates of chemical reaction. Catalysis by proximity. In order to chemically interact, substrate molecules must come within bond forming distance of one another. The higher their concentration, the more frequently they will encounter one another, and the greater will be the rate at which reaction products appear. When an enzyme binds substrate molecules at its active site, it creates a region of high local substrate concentration, one in which they are oriented in an ideal position to chemically interact. In and of itself, this proximity results in the rate enhancements of at least a 1000-fold over that observed in the absence of an enzyme. Acid-base catalysis. In addition to the contributing to ability of the active site to bind substrate, the ionizable functional groups of amino acyl side chains and where present of prosthetic groups can contribute to catalysis by acting as acids or bases. We distinguish two types of acid base catalysis. Specific acid or base catalysis refers to reactions for which the only participating acids or bases are protons or hydroxide ions. The rate of reaction thus is sensitive to changes in the concentration of protons or hydroxide ions but is independent of the concentration of other acids, proton donor or bases proton acceptors present in the solution or at the active site. Reactions whose rates are responsive to all the acids or base present are said to be subject to general acid catalysis or general base catalysis. Catalysis by strain. For catalysis of lytic reaction which involve breaking a covalent bond, enzymes typically bind the substrates into a conformation that weakens the bond targeted for cleavage through physical distortion and electric polarization. This strained conformation mimics that of transition state intermediate, a transient species that represents the midway point of transformation of substrate to products. Nobel laureate Linus Pauling was the first to suggest a role for transition state stabilization as a general mechanism by which enzymes accelerate the rates of chemical reactions. Knowledge of the transition state of an enzyme related reaction is frequently exploited by chemists to design and synthesize more effective enzyme inhibitors, called transition state analogues, as potential pharmacophores. Covalent catalysis. The process of covalent catalysis involves the formation of a covalent bond between the enzyme and one or more substrates. The modified enzyme thus becomes a reactant. Covalent catalysis provides a new reaction pathway whose activation energy is lower and the rate of reaction therefore faster than the pathways available for homogeneous solution. The chemically modified state of enzyme is, however, transient. Completion of the reaction returns the enzyme to its original unmodified state. Its role thus remains catalytic. Covalent catalysis is particularly common among enzymes that catalyze group transfer reactions. Residues on the enzyme that participate in covalent catalysis generally are cysteine or serine and occasionally histidine. Covalent catalysis often follows a ping pong mechanism, one of which is the first substrate is bound and its product released prior to the binding of the second substrate. Figure 7.4 Ping pong mechanism for transamination. ECHO and ECH2NH2 represent the enzyme pyridoxal phosphatase and phosphate and enzyme pyridoxamine complexes respectively. So the first alanine will bind and then pyruvate will go off. Then ketoglutarate will bind and then glutamate will leave off. Okay. So now coming to substrate-induced conformational changes in enzymes. When Fisher's lock-and-key model accounted for the exquisite specificity of the enzyme-substrate interactions, the implied rigidity of the enzyme's active site failed to account for the dynamic changes that accompany catalytic transformation. This drawback was addressed by Daniel Koshland's induced fit model, which states that as substrate binds to the enzyme, they induce a conformational change that is analogous to placing a hand substrate into a glove enzyme. The enzyme in turn induces reciprocal changes on its substrate, harnessing the energy of the binding to facilitate the transformation of substrate into products. The induced fit model has been amply confirmed by biophysical studies of enzyme motion during substrate binding. So, two-dimensional representation of Koshland's induced fit model of the active site of a lyase can be seen. Binding of the substrate AB induces the conformational changes in the enzyme that align catalytic residues which participate in catalysis and strain the bond between A and B, facilitating its cleavage. HIV protease illustrates acid-base catalysis. Enzymes of the aspartic protease family, which includes the digestive enzyme pepsin, the lysosomal cathepsins, and the protease produced by the human immunodeficiency virus HIV, share a common mechanism that employs to conserve aspartyl residues as acid-base catalysts. In first a stage of reaction, one aspartate functions as general base. In figure 7.6 we can see aspartate functions as a general base that extracts a proton from water molecule to make it more nucleophilic. The resulting nucleophile then attacks the electrophile carbonyl carbon of the peptide bond targeted for hydrolysis, forming a tetrahedral transition state intermediate. The second aspartate here, it then facilitates the decomposition of this tetrahedral intermediate by donating a proton to the amino group produced by rupture of peptide bond. The two active site aspartates can act simultaneously as a general base or as general acid because the immediate environment favor ionization of one but not the other. So this figure shows mechanism for catalysis by an aspartic here protease such as HIV protease. Curved arrows indicate the direction of electron movement. First, aspartate X reacts as base to activate a water molecule by abstracting a proton. Okay. Second, the activated water molecule attacks on the peptide bond, forming a transient tetrahedral intermediate. Aspartate Y acts as an acid to facilitate the breakdown of tetrahedral intermediate and release of the split products by donating a proton to newly formed amino group. Subsequently, shuttling of the proton on asp X to asp Y restores the protease to initial state. Okay, we'll continue the chapter in the next video. Like, share, subscribe.